Yeah. What's the take on the market right now in terms of other companies that are looking to develop and capitalize on circular RNA? I mean, are you one of a few, one of many?
I would
You
say,
can make him honest on this, Andrew. You cover the space. You follow the
I
space.
think
It's
it's
like, oh,
about five to
there's no one
ten.
else like me.
It's getting hotter and hotter.
It's getting hotter and hotter. I think,
Yeah.
we were first out there with these claims, and then I think many people have come along and corroborated the claims, I think, to their own satisfaction. So I think, well, we've got to get in on this action.
Hmm.
The thing is, though, that as far as I'm aware, there's only one way of making circles efficiently, and that's the way that Alex discovered in Dan's lab,
Hmm.
and we have an exclusive license to that. So now, having said that, there's more than one way to make a circle, and you can make it inefficiently if you wish. And if you, in the end, had 1 gram of circle made this way and 1 gram made that way, it's still a gram of circle. At that point, then it's what does it encode? What are the other differences in the molecule? But in terms of ease and efficiency, I would say ours is made in one step. To be clear, there's no capping, there's no tailing, there's a single enzyme, which is the polymerase. There's no capping enzyme or tailing enzyme. So there's no mods. We don't need mods because circles purify more readily from the contaminants in the in vitro transcription reaction for which you need the mods. So if you don't have the mods as contaminants, then you don't need the mods at all. And mods are much more expensive than regular nucleotides. So our cost to make a gram of finished stuff is like 1/10 to 1/15 of mRNA.
Yeah, there's some real,
Yeah.
there's some really nice supply chain benefits to working with circular RNA. And I think too, just the field talks a lot about the clinical benefits as well, or, the hoped for clinical benefits in the future, right? In terms of overcoming some of this, just the stability, the durability limitations that we've seen with linear mRNA. So I've been watching it really, I've been watching it with a lot of anticipation to see where it goes, because I have been watching sort of the numbers of pipeline products, right, start to creep up in the circular RNA space. It's getting some more respect and some more attention. And I really liked what you were saying earlier too, because it's, you were talking about it, sort of the iterative innovation, right, that the entire space is sort of going through, and how you approach failure and the challenge of working in a potential area that still has yet to really find its future, right? I think the RNA space as a whole, particularly on the encoding side, right, your mRNAs, your self-amplifying, your circular, still have a long ways to go, right? I think in terms of efficiency of development and their biological impacts, right? So I'm really curious to kind of pick your brain on how heading up a circular RNA company is challenging today, right? Especially since we are still trying to figure out, I always kind of call it the mRNA identity proposition, right? Within the advanced therapy space, within the biologic sector. How do how are you navigating some of the risks of working with a very novel technology today? And how does that impact your leadership strategy, right, of a company that's kind of been paving the pathway for this modality?
Yeah, I mean, the risks we embraced are substantially more than the ones we've been talking about, because, as I mentioned, the first thing we were meant to do was go find a lipid, a particle, which would naturally deliver to immune cells. Such a thing did not exist.
Mm-hmm.
Such a thing was never suggested to have even been possible. Everyone kind of thinks these things go to the liver as a default. So not only were we trying to develop circles as a novel payload and promulgate the idea that they don't need mods, which is iconoclastic. In fact, a Nobel Prize has been given on the need for mods, and there we are saying, well, you don't actually need them if you use circles. I'm not saying it wasn't a great idea. I myself have received linear RNA vaccines, but it's an interesting point. Technology evolves. But we're trying to do this thing with delivery as well. So we were doing two things. So, my feeling was, yeah, this is not going to work.
I love the honesty.
But, so how do you do that? Basically, it's exciting what you're trying to do. The vision is there.
Yeah.
And some people are always drawn to that exciting possibility. They're risk tolerant, I guess, like I am. That's always true in the early stages of a startup. I mean, NPM was a very good, a very solid name in terms of the venture backing. So there was good funds available. But, you also have to have a backup plan. Okay, what do you do if this doesn't if this doesn't work out? And so we had all that, but in the end, we actually got it to work. And so that was an amazing day, that we could deliver to immune cells, and they would take up the circles. The circles would express, they'd produce a CAR protein on the surface of Ts and NKs and macrophages, and then those cells would go and attack other cells in the animal, just like a CAR T does. So that was pretty amazing. And, we showed that. So our innovations were not only academically that we showed how to make, this is the broader we, of course, this is Dan's lab, but, Orna and its origin story, showed how to make circles that was possible. We showed how to express from circles. We showed that it was possible to find an immunotropic lipid. We showed the value proposition behind doing that and what you would do to get that to work. So we have blazed a whole series of trails as we've pushed into this. One of the challenges, obviously, when you start out is also that it's very hard to be noticed. And good companies being started all the time. And when I was at Intellia, it was a luxury because you're noticed. I mean, editing, everyone would notice every single thing you would do. People were lining up around the block to join the company. It's like you could pick and choose whoever you wanted to come. It was that intense. And RNA was still very, it was very hot at the time. The pandemic had started, and there was some value in the vaccines. But you still, you try and be noticed. And one of the things that was surprising, I still don't really understand to this day, is that the idea that the RNA could be circular seemed to capture the imagination of reporters or people who like to write about it. In other words, it wasn't like, oh, we've got a different cap structure, or we do mods a different way, or something like that. No, the RNA is circular. It just seemed intriguing enough that we got noticed early enough. And because we were sort of the first one out there, and because it's right there in the name, we kind of became, I don't want to say synonymous, but we certainly became like the name in the space. So it's a little bit of luck, being hot, a little bit of sort of marketing. But there was also that inexplicable, I don't know why circles capture the imagination, but they just do.
They're so poetic.
Yeah, I mean,
And your ornicorns, I mean, come on, it's the myth is just baked into your company.
There it is.
You talked about being first on the scene. It's funny. I was just thinking about an episode that dropped a few weeks ago, an episode of the Business of Biotech that I named The Innovator's Dilemma. It was with a friend of mine, Brian Finrow at Lumen Biosciences. And we talked about that concept of being first on the scene with a new technology or a new approach, and how it creates awareness, potentially inherently, right? It's something new, but it can also create tension. It can create challenges around getting people to spend money on you. You've mentioned NPM a few times. It's fantastic to have a good strong partner there. But I guess take us behind the scenes a little bit around when you're revealing something new to the community, not just the scientific community, but the investment community. What goes into ensuring that you're winning the funding you're going to need to move forward and hit milestones?
Yeah, I think it has to be the logic of the idea. I might even say the logical ineluctability of the idea. The premise here was that cell therapy is fantastic from an efficacy perspective, but it's so cumbersome, so difficult, so challenging for patients. They have to be managed so intensively that there's the cost of the therapy, and then there's the adjunct costs of just administering and managing the patient during the administration of the therapy and the risk associated with that. And so the idea was, well, if you could just do the manufacturing inside the patient, not at the bedside, actually just in the patient, that would make everything go away. And that's self-evidently true. The idea that if only you could, then that would be great. That's an easy sell because that's obviously true. The question is, well, how the heck do you do that?
Right.
At least our attempt, which in some ways didn't have to work, but it was a shot on goal. But there was no name for what we're trying to do. Now the field is called in vivo CAR or in vivo CAR T. So there are some groups that are trying to do it through adapted viruses, where the viruses will do the job of delivering. Now, of course, they tend to then integrate into the genome. Some viruses don't. but like lentivirus vectors will integrate. And ours is transient, but it's redosable, which a lentivirus is not. So when you get to that point of showing that it works, the value proposition is self-evident. Okay, it's like, yeah, if only you could do this, like teleportation. Yeah, that could be useful. Okay, can you do it? And so if you do a little demo, I don't have to explain what you would do with teleportation. You can take, your mind can take it from there. So I just have to show you, look, particle, animal, boom, cell's gone, tumor gone. People go, I like. Here's money. Yeah.
here's money. Matt, we really picked the wrong career.
I feel like I picked the wrong question. Dr. Barnes's answer was like, well, because it works, dummy. Hey, people.
You're millions of dollars.
Here you go. Yeah, it's interesting too. Like sometimes I have a hard time. My mind, I'm a slow Pennsylvania boy, right? Like my mind doesn't keep up. Like the you mentioned earlier the prospect of sort of leapfrogging RT, and that just kind of, to me, that's kind of mind blowing. Like, you know, CAR T is still new, and if you talk to CAR T people, they're telling you like, oh, we haven't yet scratched the surface of the indications that we can tackle. Yeah. Right. And we're making great progress in solid tumors and so on and so forth. So I'm curious about your perspective of how the CAR T community might, I guess, receive Orna's news that Orna thinks it can kind of leapfrog CAR T, and specifically how would Dr. Carl June feel about the prospect?
Well, our Series A was interesting because every new investor, we had venture investors up until that point, but every new investor we had in the Series A was a pharmaceutical company that was doing cell therapy.
Interesting.
Yeah. So in other words, they were saying, we too see the value proposition, and we wish to be close to this story in case it works out. So I would say embracing the potential, and obviously in some ways it's a competitive technology to CAR T, but and it's much more pharmaceutical in a way. Whereas CAR T is a living drug, it will expand. You can't really control it. Whereas this, you can control the dose. It has a half-life. It has a maximum concentration, which then fades over time. And that's a function of the dose that you give. So this is much more like a traditional pharmaceutical in that way. And so there's CAR T companies that, of course, want to defend their turf and their franchises. But there's also many things about this that would be appealing to a pharma player that isn't in cell therapy, because those who might find cell therapy a bit fiddly would probably find LNPs, whilst more fiddly than a small molecule, a lot less fiddly than a cell therapy.
I think that's an interesting point too, because we live therapeutically within the ATMP space right now, right? And so everything's kind of been marketed or positioned as this one and done, right? Which I think can have sort of a sexy, a bit of allure, right? Especially for a therapeutic that can be very expensive, right? The prospect of a durable, potential cure, right? It's not everything approved has been curative, but that one and done does have some appeal to it. I'm curious, but the fact that I think too, investors can be aware of if RNA is a chronically dosed therapy that fits similar to a biologic, right? That risk proposition is understood, right? It's well known. But I'm curious, do you find that navigating the funding today has changed at all, right? I mean, as more players come into the space with RNA, as the space, we're not mature by any sense of the word, right? But as we have moved beyond the pandemic realm, are you finding that, investors are interested in different types of information or data, or, has getting funds become more challenging in any way?
It's an interesting question. I think, as a company progresses through its stages, as its own story advances, its pipeline advances, then what you want from or need from the funding and the scale of funding you need, it inherently changes. So you're not appealing to the same necessarily. You start with a venture appeal, And then later on, you're appealing to different kind of later stage investors, and ultimately pre-public investors, and ultimately public investors. And these investors are all looking for different things, different proof points. They typically, you can probably distinguish them based on their own risk tolerance profile, where they feel their sweet spot is, and where they like to get in. In terms of our story, our story resonates, but in the case of Orna, it resonates pretty well. What we're trying to do is relatively differentiated. So it's not just the name that people remember, but they do tend to recall that we're doing what we call now pan-CAR, because our lipids don't just deliver to T cells. So if you have a lentivirus, it's going to find the T cells because it's a T cell tropic virus. Whereas our particles are pan-lineage. They go to Ts, NKs, and macrophages. And so to the point that people feel cell therapy have only scratched the surface, yes, you have some people doing CAR Ts, you have some people doing CAR NKs, you have whole companies doing CAR Max, CAR Ms.
Yep.
Well, we do all three with one injection. We create all three with one injection. So that's why we call it pan-CAR rather than, and we call it in vivo CAR rather than in vivo CAR T, because we're reaching all the effector lineages in the immune system. So, people like that. And the potential that we could do something in solids is there without having to change anything of what we do, because we're in the macrophage already. We just have to think about what the target is. What does the CAR target?
Yeah.
But there are companies out there already trying to blaze a trail in CAR Max space. We're content right now to be looking at relatively tried and true targets. This is one of the I think rules of bringing new technology forward is you don't want to stack your risk. You don't want to try new biology and new technology. So our technology is new, so we are using a fairly vanilla target.
Okay.
Helps us compare and contrast, but it also helps everybody understand what you have in that way. So
That's an interesting point.